Hello guys, today I'll be solving some topical past paper 1 type questions of CIE A-Level Biology, subject code 9700, and today I'll be solving questions from chapter 2, Biological Molecules. I've skipped chapter 1 for now because it involves a lot of questions that requires measuring pictures and specimens, and it is not quite possible to get the exact measurement through the screen and the PDF version of the past paper. That is why I'm doing biological molecules for now. So let's move on and get started. So for question number 1, which levels of protein structure are demonstrated by a hemoglobin molecule? So for hemoglobin molecule, what we need to understand is that it has four polypeptides, one alpha chain, not one. It has two beta chains and two alpha chains. When a protein has more than one polypeptide, then it demonstrates a quaternary structure, right? And to reach the quaternary structure, it must have a primary structure. So primary structure is the sequence and number of amino acids in any kind of protein. Secondary structure is when the hydrogen bonding between the amino acids in the protein causes the protein to either form a helical shape or a beta pleated sheet kind of shape. And tertiary structure is the 3D shape of the protein. And the quaternary structure, it only achieved when you have more than one polypeptide. So in this case, because all of the options A, B, and C have crossed out the quaternary structure option, the answer is D, because hemoglobin molecule demonstrates all levels of protein structure because it has 4 polypeptide. Question number 2, the diagram shows a molecule, as we can see, this molecule. So to recognize this molecule, it is very simple because it has this carbon in middle. It is also known as the alpha carbon, and this carbon is attached to one hydrogen group, one NH2 group, which is also known as the amine group, and one COOH group. This group is also known as the carboxyl group, and this is a variable group, so it changes. It's also known as the side chain or the R group, and that changes. So molecule shows a basic structure of an amino acid. So which test on polymer of this molecule would give a positive result? So amino acids are the monomers of proteins. So to give a positive result, it must be something that is testing for a protein. Adding burette solution is the correct option here because that is how we do a protein test. So for this, your answer is going to be A. Adding iodine in potassium iodide solution, that is a test for starch. Heating with Benedict solution, it is a test for monosaccharides, disaccharides, and or reducing sugar in general. And shaking with ethanol, then water, that's a test for fats, lipids, and triglycerides. So none of this would give a positive test if it was tested for the polymer of this molecule. This molecule is an amino acid, so the test by adding burette solution would give a positive result in this one. Now, moving on to number 3, which property of water minimizes temperature changes in all cells and organisms? So for this, let's go through all of the options and see what that property of water does. So cohesion is the property of water by which it is attracted to other water molecules. So it's attraction between similar objects. Heat of vaporization is the amount of heat that is required to change water from a liquid state to a gas state. And maximum density at 4 degrees Celsius is the property that water has the most density at 4 degrees Celsius. All of these properties that I've discussed so far do nothing to minimize temperature changes in cells and organisms. And so the option cannot be A, cannot be B, and cannot be C because they are not related to temperature by any means. You could be confused that it could be heat of vaporization, but that does not affect the temperature in cells and organisms because it is the amount of heat that is required to change water from liquid to gas. So specific heat capacity is the amount of heat that is required to raise the temperature of water by a degree Celsius. So because water has high specific heat capacity, it does not easily change its temperature. So that is the property that minimizes temperature changes in cells and organisms. And that's your answer, number D. Now moving on to question number 4. So, okay, this the graph shows the effect of pH on the structure of a protein which consists entirely of repeating residues of one amino acid. So that means that it is a protein structure with just one amino acid, and which statement is true. So as we can see, like it has an alpha helix shape. So alpha helix shape denotes a secondary structure because as we have discussed earlier, a secondary structure of a protein includes either alpha helix or a beta plated sheet. So alpha helix is the secondary structure, and after pH 6, it is denaturing and it is becoming a random coil. As you can see through this graph, it's denaturing and it's becoming a random coil. It's losing its shape. So which of the statement is true? At pH 2, the protein has lost its secondary structure. And that's not true because at pH 2, which is this pH, it still has its alpha helix shape. So that means it has not lost its secondary structure. At pH 2, the protein has lost its tertiary structure. That's also not true because the protein, there's nothing to show that the protein is in a tertiary structure, so that option also cancels out. So you can cancel out A and cancel out B. So at pH 10, the protein has lost its primary structure. It has not. The change in pH definitely does not affect the primary structure of the protein. So the correct answer must be D. At pH 10, the protein has lost its secondary structure. As we can see, at pH 10, it is just a random coil. It is not alpha helix nor a beta plated sheet. So the answer for this question is D. Now moving on to number 5, which types of bonds hold the tertiary structure of a protein together? So you have to remember that a primary structure of a protein is primarily made of covalent bonds known as peptide bonds. And the secondary structure has one more extra bonding known as the hydrogen bonding. But for the tertiary structure, so you can just cancel out, first let's cancel out the options and deal with the remaining option. It cannot be glycosidic bond because it's a protein and proteins do not contain glycosidic bonds. Glycosidic bonds are only contained in polysaccharides or disaccharides. When two glucose molecules or like hexose or monosaccharides are joined together, that's when glycosidic bonds are present. So it's not present in a protein molecule. So you can cancel out A and C. And moving on, it's not B because peptide bond, like I have discussed earlier, it is the covalent bond that joins and maintains the primary structure. So it has nothing to do with the tertiary structure. I mean, without it, the protein would not have its primary structure at the first place. So it would not get to this tertiary structure. You know what I mean? Because first it has to have a primary structure, then a secondary structure, and then finally a tertiary structure. So answer is also not B. And so the remaining answer is D. So that's the correct answer for this question because now you can either memorize what bonds hold the tertiary structure together, like disulfide bond, hydrogen bond, and ionic bond. Those are involved in holding together the tertiary structure, or you can just cancel out reason like that and be sure that your answer is correct. And that's how you get to the answer. And the answer for this number is D. Question 6. Which molecule in the key is sucrose? So to understand what a sucrose is it a reducing sugar? Sucrose is not a reducing sugar. So there are in biological molecules in chapter 2, you have to study about 3 monosaccharides, hexose, galactose, and fructose. And All of them are reducing sugars, and those sugars are involved in forming disaccharides like maltose, lactose, and sucrose. Among these disaccharides, maltose and lactose are all reducing sugar, but sucrose is the only disaccharide that you have to study about. It's not the only disaccharide, but that's the only disaccharide that you have to study about. That is not a reducing sugar. So you go on to this pathway. This is not an option anymore because it is not a reducing sugar at all. So does it contain hexose sugar? So hexose sugar are those sugars that have 6 carbon atoms, and they commonly include like glucose, galactose, fructose. All of them are hexose sugar. So sucrose is made up of, let me just type it out. So sucrose is made up of glucose and fructose. Both of them are hexose sugar. So it does contain uh, hexose sugar. So it is not a reducing sugar, but it contains hexose sugar. So the correct answer for this would be C. Okay, so for question number 7, bonds are formed by condensation in cellulose, sucrose, and hemoglobin. What are the name of these bonds? So condensation reaction, they always form a covalent bond. So first you have to recognize what these molecules are and what are their monomers, and from there you can identify what type of bonds do they have. Cellulose is a polymer of beta glucose. So because it has glucose, it consists of glycosidic bond. So cellulose must have glycosidic bond, so that means C and D are not the option. Sucrose is also made from glucose and fructose, so it also involves monosaccharides. So it A is not an option because the bond present in sucrose would also be glycosidic, and hemoglobin is a protein, and proteins have peptide bond in them. So the answer for question 7 is option B. As you can see, hemoglobin, it cannot have ester bond. Ester bonds are present in lipids, fats, and triglycerides or phosphoglycerides. And there is another bond known as phosphodiester bond, which is present in DNA molecules, nucleotides. What will break an ionic bond between amino acids? So ionic bonds, they are a very weak kind of bond, and they are not strong enough like a covalent bond, so they can easily be broken down. But then we have to consider the options here. Condensation is the type of reaction that forms a bond, so it will not break a bond, but a condensation forms a covalent bond, and hydrolysis is the reverse of condensation reaction. So it uses water and breaks the covalent bond between amino acids or any other molecules. So hydrolysis is also not an option here because it's breaking a covalent bond, and what we are asked, what will break an ionic bonding? Low temperature also does not affect ionic bond, because so the answer will be D, pH change, because the change in pH can affect the charge of an amino acid and change its or break its ionic bonding. But now, moving on to question number 9. The diagram shows a part of a polymer. Which molecule is used to break the bond that is indicated by the arrow? So you might get easily confused here. I've chosen this question because it can be confusing in a way that it is phrased. Because the arrow is pointing to a bond between carbon and nitrogen, you might think that they are asking for what kind of bond it is. And because peptide is the answer, many students get confused and choose peptide because most of them don't read the question when they are answering MCQs, and that's a very big problem. You have to read through the question properly, because just because it is pointing to the peptide bond does not mean that the answer that the question requires is peptide. So because the question is asking which molecule is used to break the bond indicated, answer cannot be peptide because peptide is the bond that is indicated in here. And how can you recognize what a peptide bond is? It's a bond between a carbon and a nitrogen atom, and it's sharing of electrons, and it is present in amino acids. As I have discussed about the basic structure of amino acid before, you can keep in mind. So here, as okay, I'll show here this is the alpha carbon in one amino acid, and this is the alpha carbon in another. Here is the H group, that is hydrogen group, that is present in both, and CO, and here was the OH group, and here is the carboxyl group of it, and this one is the amine group of one and amine group of another. So covalent bond is formed when H from the amine part of one amino acid and the OH from the carboxyl group of another amino acid, they are joined together and water is released, and this bridge is formed between carbon and nitrogen. So amino acid is not the molecule that is required to break the bond because it forms a part of the polymer. It does not break any bond or form any bond. Amylase is an enzyme, so that's also not our answer, and the answer here is water, and I'll also explain why. So because the amino acid, this part of a polymer, it shows a peptide bond. It is formed by a condensation reaction which releases water, and to reverse that reaction we require water. In the hydrolysis reaction, the breaking bond releases two amino acids, and this breakdown happens in the presence of water. It is the basis of hydrolysis reaction, and because the word hydrolysis has the part hydro in it, and lysis, hydro means water and lysis means break apart, so hydrolysis, using water to break apart the bond. So that's how you can remember if there is any reaction like hydrolysis reaction, it uses water to break the covalent bond between monomers. That's your answer for question number 9. Answer is D. Question number 10, probably the last question of today, is I have a lot of questions. There are like 12 pages in this document, but then because I'm explaining most of them in detail, it's taking a lot of time. So I'll probably upload these videos in parts. So please look forward to more topical questions on biological molecules. So here, this is one very confusing kind of question. Um, so four sugar solutions were tested with the standard Benedict solution. So whenever there is a Benedict solution, they are testing for a reducing sugar. The table shows the color of the solutions after being tested. Whenever reducing sugar is tested with Benedict solution, the color of the reaction changes from blue because the Benedict solution is blue in color. So when you add it to a sample, the color of the sample also turns blue. So if the reducing sugar is present, it changes from blue to green to dark green to orange to brown to brick red. Okay, so this is how the color progresses in sample contains reducing sugar. So if it is treated with Benedict solution, then the color changes from blue to green to dark green to orange, brown, and brick red. So what is the best interpretation of the results? So solution 1 has green color, so it has some reducing sugar present, not a lot. This progression also shows quantitative result of how much. It does not give a exact value, but it gives you an idea of how much um, reducing sugar or non-reducing sugar is present in a solution. Non-reducing sugar, it only tells you when you treat the sample first with hydrochloric acid and then finally use Benedict solution to treat it. So yeah. So because solution 1 is green in color, it has some reducing sugar. Number 2 is blue, so it has not changed colors. The sample has not changed color at all, so it means that reducing sugar is not present, but non-reducing sugar might be present. Solution 3 has brick red color, so that means that it has reducing sugar and has the most amount of reducing sugar. And solution 4 is yellow, so it has more than 1 and less than 3. So solution 4, let's look at the options. So we can cancel out option B and option D because the solution 2 does not have any reducing sugar. So two of them are cancelled out, and brick red, it has the most amount of reducing sugar. So option 1 and 2, it makes sense. 1 has some, but it has more than 1. One has, in option C, solution 1 has 1% reducing sugar, but solution 4 has only 0.5% of reducing sugar, when solution 4 must have more than 1 and less than 3. So option C is also not correct because solution 4 must have more reducing sugar than 1 and less than 3. It has less than 3, but then it has less than 1 as well, so that's not the option. So your correct answer will be option 1 because it has solution 2 has non-reducing sugar, so it is blue. Solution 1 has some reducing sugar, so it is just green. Solution 3 has the most percentage of reducing sugar, so it is brick red. And solution 4 has less than 3, 0.1 is less than 3, but it has more than solution 1, 0.05 is less than 0.1. So solution 4 has more percentage of reducing sugar than more concentration of reducing sugar than solution 1. So your option is 10A.